welcome our president, Kamlesh Bhai, and our speaker for today. I would also like to welcome our speaker for today, Ms. Kinjan Parikh. As everyone is aware, FPME sets a theme for every new financial year. For the upcoming year, our theme will be awareness and networking. Awareness to introduce FPME to various government regulatory bodies and present our concern related to export policies. Networking to add more merchant exporters from different parts of the country to strengthen FPME. To keep our members updated, we have annual calendars of webinars and fireside chat to discuss and make a roadmap for exports. Today's topic for webinar is in continuation to the last month's webinar. Today, we will understand different regulatory markets and their requirement for submission of dossiers. Let us begin with the webinar. We will have Q&A after the session. I would request the members to please sit through the session and post your questions at the end. Our speaker for today is Ms. Kinjan Parikh, founder of Achievers Regulatory Services, Ahmedabad. She is M.Pharm in Regulatory Affairs and Management. Ms. Parikh has successfully scaled up her company in a span of 3 years by registering 10 plus dossiers in various regulatory markets. I request Ms. Kinjan to please start with the webinar.
Good afternoon, everyone. Thank you, Jyotika ma'am, for such a good introduction. I would also like to thank FPME for conducting such a webinar and for such important topics that is related to export of our medicines. And this cannot be happened without guidance of from the ख्याति group of companies. So we will start our presentation today. As is it? Can everybody see? Is it visible? Can I start now?
Yes, you can, Kinjan.
Yes. So today, as Jyotika ma'am already said, our topic is on different pharma regulatory markets for dossier registration. We will cover overview of different regulatory markets and the requirement for submission of a dossier to different regulatory authority, different pharmaceutical guidelines. What are the requirements for the dossier registration in different countries? So today we will talk about what is regulatory authority, what are the types of regulatory market, what are the general regulatory process to file a dossier. Also, we will see what are the different countries and the respective regulatory authority name. What are the general requirements for registration of dossier? Stability, different stability testing conditions, labeling statements for FPP product. Also, we will cover two very important topics that are required for submission of a dossier, that are BA/BE studies and BCS classification, and what is BCS BioWaiver studies. So starting with what is pharma regulatory authority. Whenever we are going to export our product to any of the country, each of the country has its own regulatory authority. And the main aim of the regulatory authority is having a stringent drug approval system to ensure that our medicinal product that we are going to export or that we are to launch that are manufactured with acceptable quality and efficacy. And each of the country has its own guidelines, own regulatory guidelines through which we need to follow its regulatory process. And that has to be followed by any of the every person, either it can be an organization, a sponsor, or innovator. Any of them has to follow that guidelines that are mentioned in their website or that are listed in that state website. We need to follow each one of them, and then they give an approval to launch our product in that country. And this process is known as drug approval process. And each regulatory authorities has different guidelines for different submissions, for submission of different pharmaceutical forms, veterinary form or biological form. Here pharmaceutical, that means we have different guidelines for each form. For herbal medicine also we have different guidelines. Medicinal devices also have different. For veterinary and biological food, here biological food means a vaccine or serum or any type of that derived product. Each one of them has its own different guidelines, and we need to compile with each one of them to submit our brochures. Now, what are the type of regulated market? Here it is divided into one, that is regulated market. Regulated markets means here it involves those countries which have a defined regulatory requirements set by regulatory bodies of the country. That means they have a strict regulatory guidelines that we need to comply to submit or to approve our dossier there. Here regulatory, that means we cannot leave any of the list of documents that are mentioned in their guideline. We need to comply with each one of them, and that only they will evaluate our dossier, and then we can give it. Then we are given approval. Here, regulatory market cover countries like US, EU, Canada, Australia, New Zealand, South Africa, Japan. EU countries means countries like UK, Germany, France, Ireland, Sweden, etc. And second is semi-regulated markets. Semi-regulated markets are those that who still lag behind putting forward the well-defined definition for drugs and have their own regulatory compiling with regulated market guidelines. That means, of course, they compile with regulated market guideline, but they are not as stringent as regulated market. Means they have their own some inclusion and exclusions, and they are not that strict as regulated market. Means if regulated market require 3 batch of certificate analysis, semi-regulated can have only one, can ask for only one. That means they are not as strict as regulated. These are known as semi-regulated market. And the regions that comes under semi-regulated market are Asia-specific countries, Latin American countries, Eastern European, African, and Gulf countries. Here, Asia-specific countries are grouped into a name called Asian nations, Southeast Asian nations, and Gulf countries are grouped into an organization named as Gulf Cooperation Council. And the regulated market is those countries that don't come in regulated market. The another name is ROW country, that is rest of all countries. Here they are subdivided into Asian countries, African, Asia, divided into continents like Asia, Africa, Middle East, Latin, Francophone, and GCC. Then CIS. Asia consists of countries like Sri Lanka, all that like Sri Lanka, India, Bangladesh, and also it consists of Asian countries. Asian countries, that is a group of 10 countries, namely Philippines, Vietnam, Singapore, Malaysia, Thailand, Indonesia, LOAS, Cambodia, and Myanmar, and Brunei Darussalam. African countries are Algeria, Zambia, Ethiopia. Here, African countries consist of every part of the West Africa, South Africa, East Africa, every one of them. Middle East countries are like Gulf GCC countries like Bahrain, Kuwait, Oman, Qatar, Saudi Arabia, and UAE. Latin American countries are like Mexico, Brazil, Panama, Peru, Guatemala, Argentina, Chile, and Dominican Republic. Here, Latin American countries have their own set of regulatory guidelines that are not stringent as regulated or semi-regulated. CIS, that is Commonwealth of Independent States. Here also they have limited. They require only limited set of documentation for registration of dossier. They don't have that lengthy type of documentation or very extensive type of they don't require that. They only require limited type of documentation, and they are available from their MOH site. And that is listed in their MOH. Francophone countries consist of Congo, Ivory Coast, Niger, Mali, Senegal, Madagascar, Rwanda, Togo, and etc. Here are the general regulatory filing process. Now, when we are exporting our product, then we need to follow. Then we need to know what is a general regulatory filing process to file our dossier to any of the country that we need to export. So, starting from the very first step, that whenever we need to develop a product, and export our product, first of all, we will finalize that whatever API will be, that means what the manufacturer of the API will be, what will be the manufacturer and what will be the vendor of our excipients that we are going to use in our formulation, and what are the type of packaging material we will require for our packaging of the formulation. So first of all, we need to finalize what are the vendors for excipients and packaging material, and what will be our API vendors name. And very importantly, it has to be the API manufacturing site should compulsory should be WHO GMP certified, and it should have a valid GMP WHO GMP certification. Without API manufacturing site having valid GMP certification, no regulatory authority will approve our product. So after finalizing each one of API, excipient and pharmaceutical packaging material, we will move forward to our finished product development. That is our finished product that we are going to export. Finished product develop after developing after our product is developed, we need to further go for bioequivalence study and clinical study that are carried by CRO. Here, bioequivalence, what are bioequivalence study, we will talk in the later part of our presentation. And clinical studies are only important if our product is a new drug. For generic product application, clinical studies are not required. For after having all the data from our pharmaceutical development stage and from the bioequivalence study data, we will compile our dossier according to the guidelines of the country that we are going to export. For example, if we are going to export our product in Tanzania, we need to comply all the format. We need to compile our dossier in a format that is given in the TMDA site. We need to compile the for dossier in either CTD format or ACTD format that we have already, and what are these that we are already covered in our previous webinar. So we need to compile our dossier according to the format given in the respective country guidelines. After our dossier compilation, we will go for our dossier submission to the respective regulatory authority. After our dossier is compiled, we will submit to the regulatory authority. That regulatory authority will evaluate our dossier. The normal evaluation time is from 3 to 6 months. Either it can take a year also. During that evaluation, our product can be either approved or the regulatory authority will give our query, official query letter. We need to address all the query in a query response, and we need to give a query response of their given query. They will evaluate, and if they are satisfied, they will give a drug approval and our product will be registered, or either they will give another time for the re-evaluation of our dossier. And between that, if our manufacturing site is not inspected, they will give we need to give a GMP inspection certificate inspections invitation to the respective regulatory authority. Regulatory authority will come here, they will inspect our manufacturing site, will give its approval, and meanwhile our dossier will get evaluated. If both of them complies with their guidelines and their requirements, then finally our product will get dossier will get approval and our product will get registered. So these are the normal general filing procedure that is followed to launch our product and to get our product registered. These are some list of the countries and the regulatory authority. Please note this doesn't cover each and every country and name of each and every regulatory authority. These are some common countries and it's a name of common regulatory countries, and these are given in alphabetical order. Some of them are missing, but these are just for our normal to go through or the how our countries and how they are and what they are regulatory name. So some of the regulated market regulatory names are starting from the first is Australia, that is therapeutic, that is TGA. Another regulatory market is regulated market comes from Europe, that is European Medicines Agency, EMEA. These both comes under regulated market. Also Canada had its regulatory authority name Health Canada. So these are some of the regulated market and the name of its regulatory authority. The second slide consists from the I to N, and here the second row is India. A regulatory authority name of India is Central Drug Standard Corporation Organization, that is CDSCO. In this slide, you can see that some of the countries have only regulatory authority name as Ministry of Health, that is MOH. Means it is not necessary that each country has its name, the particular individual name of its regulatory authority. It can only have its Ministry of Health, and we need to comply the guidelines that are given in their Ministry of Health website. And the last one is from P to Z, and some of the, and here it covers USA, that is regulated market, and the name of your regulatory authority of USA is Food and Drug Administration. Now the main part here, what are the general requirements for the registration of dossier in regulated or semi-regulated market, in regulated market? In our previous webinar, we talked about what is CTD dossier and what is ACTD dossier, what is API part and what is our finished product part, that is known as drug substance and drug product respectively. So we all know that what is CTD and CTD. CTD consists of 5 modules and ACTD consists of 4 modules. So they are administrative documents. These are the documents that comes under part 1 and module 1 of ACTD and CTD respectively. These are administrative documents. That means for any country, some of the common administrative documents that you need to submit are COPP, that is Certificate of Pharmaceutical Product Permission, Manufacturing License, WHO GMP certificate, pre-sale certificate, and artwork. These are some of the common administrative documents that we need to submit in part 1 or module 1 of our dossier. Here, some countries require COPP, product permission, manufacturing license, legalized. That has to be notarized. And while few countries don't require the same, we need to just add the soft copy of the same. These are documents, common documents required in part 1 or module 1 of ACTD or CTD documents. Now, second is what are the documents that should be majorly covered in the API part, that is DMF part of our dossier. API part should consist of normal common factors, common documents like starting from its nomenclature to general properties, manufacturing, manufacturer and manufacturing site address. Both site of manufacture means site address of the API manufacturer should comply with that given in the GMP of the API manufacturer. Means whatever the address that is given in GMP, the same should be written in the manufacturer part of the DMF also. It should have an elaborate route of synthesis with its flow diagram. Structural elucidation is also important. That means by which method we have confirming that our product is the same. Impurities should be mentioned. Impurities should be mentioned depending upon our product is pharmacopoeia official in-house. If our in-house we need to list each one of them that we have calculated, and if it is pharmacopoeia official, we need to list each of the impurities that are listed in respective USP or BP monograph. Specification and method of analysis. We need to give specification and method of analysis of our API that should comply with a pharmacopoeia standard. And container closure system, that is we need to give spec, MO and COI, each one of them, of our primary packaging material and secondary packaging material. And stability conditions, stability data. That means 3 consecutive batch stability data should be given. And what are the stability conditions that need to be followed should be according to the country that we are exporting. That means WHO has divided the stability conditions into 4 zones. Then we need to first check that the country in which we are exporting falls under which zone. We need to confirm the temperature conditions of that particular zone, and then we need to conduct our stability data according to that temperature zone, and we need to perform 3 batch accelerated and 3-batch long-term stability data of our API, and then submit the stability data and COA of API of the applicant. That means, of course, the API manufacturer will give the certificate of analysis, but FPP manufacturer has to also give 3-batch API certificate of analysis. And last, and the one of the another important class is close path. Close path consists of documents like control of raw materials, critical steps, manufacturing development, and PVPD studies. Here, and this close path is not necessary for each and every country. The country which require close path are the African countries and Asian countries and regulated countries. Some of the CIS, GCC countries don't require close path. So close path is only compulsory in Asian, African, and regulated countries. Now moving forward to FP part, we talked about what are requirements for the API part, and now what are the requirements, technical documents required for the FP part, that is our finished product part. So starting from the manufacturing process or manufacturing part, so we need to give each and every detail of the manufacturing process. We need to give each and every detail in our manufacturing process. That means what are the raw materials that are used for our manufacture for the manufacturing of our product, what are the equipments we have used for our manufacturing, and also we need to list that what are the capacity of the equipment that we have used, then what are the process parameters that we have taken care of for manufacturing of our process parameter. Here process parameters means what are the time, that means for how much time we have mixed or centrifuge, what are the temperature that we have taken care of, what is the pH of our final solution or the semi product, then what are the environmental conditions we have taken care of. Each one of them has to be addressed in our manufacturing process. And also flow diagram, description of input. Yes, description of in process controls means during our manufacturing we see the description, we see the pH of our solution of our product. So we need to describe each of the process parameters also in our manufacturing process. And flow diagram, here flow diagram is also very important, and it must consist of each of the API or excipients we have used in our manufacturing process, starting from the dispensing to the end packaging. Everything should be listed in flow diagram with the in process parameters that we have used must also be added in the flow diagram. Master formula. Here master formula should consist of the overages unit formula and batch calculation and our batch size calculation. If our product for example is any vitamin product, then of course it will have overages, right? So we need to give the what percentage of overages we have added in our formulation, and we also need to give justification that why this the amount of overages we have added. And we also need to give unit formula. That means if our product is tablet or capsule, we need to give formula for each tablet, and for if our product is suspension or syrup, then for each ml or 5 ml or whatever our dosage form, what dosage is. And also for the batch size, we also need to give batch size calculation. For example, if our product is for 1 lakh tablet, a batch size is for 1 lakh tablets or capsule, we also need to give batch calculation of the same. Now BMR is batch manufacturing record. That means BMR, we need to give master BMR and executive BMR of our finished product. BMR is again not compulsory in every country. Depending upon the regulatory guideline, we need to first check the regulatory guideline of the respective country, and we need to submit accordingly. Process validation protocol and report. That means PVP and PVR. Whatever the process validation we have carried out, and we need to submit 3 batch process validation protocol and report for every country. This is compulsory for every market. And 3 batches should be of same size, similar to the same batch size mentioned above in manufacturing formula. That means the batch size will be same that is listed in manufacturing formula and that we have taken in for process validations also. Here, the last point is formulation and development. That means PDR, product development report. Product development report consists of either a few countries ask for extensive product development report and few don't ask for the extensive. Here, extensive product development report means it also consists of all the trial information, all the compatibility studies between API and excipients, and of to our finished product, and also stability comparisons and some technical comparisons between our innovator product and our test product. So extensive PDR is only required whenever is only submitted if the country asks for it. And these are the countries, some few countries that ask for the extensive PDR are Russia, Ukraine, Algeria, Kenya, and few of the Asian countries. Now we have talked about what are requirements for API and FP part, but excipients are equally important. Excipients can be whichever excipients which are using for whichever formulation, either if it's tablet, capsule, syrup, suspension, injection, whatever it is, our excipient must comply with USP or BP pharmacopoeia standard. If the excipients doesn't comply, then if it is in-house, then we need to give vendor COA. And few countries also ask for the latest pharmacopoeia, latest monograph of the pharmacopoeia, whichever pharmacopoeia standard we have used. They ask for the latest monograph of the same. We need to add the same in our pharmacopoeia, in our dossier. And also few countries ask here few countries can go with normal USP BP monograph, that is official monograph, they can do with that. But some countries ask for the different ask for the spec MO and COA of excipients from the FPP manufacturer also. So we need to make spec MO and COA complying with the pharmacopoeia standard in the name of our FPP manufacturer. And Q&Q for the in-house powder coat mix excipient should also be given. Yes, means our product is tablet, then means it is film coated tablet. Either we have coated our tablet by taking individual coating materials, or either we have directly purchased a direct coat mix of the that for the coating. So if you have purchased already coated mix, then we need to give Q&Q, and that has to be given from the vendor excipient. Means from whichever the excipients we have purchased our readily coat mixer, they need to give the Q&Q of the this coating mixture, and that we need to submit and attach in our dossier. That is non-animal origin certificate. That means the finished product. That means we need to give a certification that our finished product means whatever excipients we have used in our finished product, none of the excipients are derived from any animal or human source. And okay, so second is finished product specification and method of analysis. This comes under control of drug substance part. Till now we have covered about what are the manufacturing, what are the requirements in our manufacturer part, what are the requirements in our excipients part. Now what are the requirements in a control of drug substance part. This consists of finished product specification, method of analysis, validation, and batch analysis. So finished product specification method of analysis, that means if our product is pharmacopoeia official, then we need to comply the each and every test that are listed in the respective monograph. But if not, if our product is in-house, then we need to comply as per the test or format given in ICH guideline, ICH Q6A guideline. And method of analysis, that means method of analysis of each of the tests that are listed in specification has to be given. And if based on pharmacopoeia additional product related specifications should be included as in-house specification. For example, description, hardness, friability, average weight, dimension, identification of correlars, and that means obviously means if our product is for example a tablet, then tablet and our tablet is official in pharmacopoeia, they will not cover not this additional document additional tests like hardness, viability, and average weight. So with all the tests, with all the impurity and description tests that are given in pharmacopoeia, we need to add our in-house test also, like what is our average weight, what are the friability, and what are the hardness of our tablet, and what if we are adding the color for our coating material, then we need to also add the identification of the coloring. And of course, if our we need to add the microbial test, if not given, if it's a capsule, syrup, or suspension, or any type of such dosage form, we need to also have the MLT, that is microbial limit test, in our specification. And sometimes what happens that the product is officially in both BP monograph and USP monograph, but they won't have same list of tests and differ in one or two tests. So sometimes we add a test from the another pharmacopoeia standard in our specification. For that, we need to give a copy of that monograph. That means that we have taken a test from this monograph in our specification. And any other test that we have included in our specification has to be addressed. That why and from, that means if it is in-house or if it is BP or we have taken from USP. Last is the limit of in-house related substance test should be given. Here it is like if our product in-house, then we must perform the related stability test or related substance test or inorganic impurity test. We need to compulsory add as test in our specification, finished product specification, and it must in our specification. But we have performed by inorganic method, but what will be the limit? How we can choose that limit of our finished product in-house test? Then for that we need to comply with the guidelines. I forgot to mention that in the slide. You can refer ICH guideline Q3AR2. This is a guideline for impurities in new drug substance. That means they have given the what should be the limit that we need to add of according to our dosage of our finished product. So we need to comply with the ICH guideline Q3AR2, Q3BR2, and for elemental impurities ICH Q3DR1. These are some of the ICH guidelines that we need to check before giving the limits of our in-house related substance test. Now coming forward to batch analysis. Batch analysis, how many batch certificate of analysis that we need to submit will be we need to choose from the that mentioned country guidelines, and it should be preferably of the batch of which the sample will be submitted for registration. Means some countries also ask for the sample before our product is registered to evaluate our sample. So the batch analysis certificate of analysis should be submitted on the same method of the same batch that has to be we need to submit for our registration. And then comes method validation for FP and API. That means if our product is pharmacopoeia official, we need to only give verification. That means it only comes on the limited test for the validation. And if our product is in-house, we need to give validation for each and every validation parameter. But few countries like countries like Asian countries ask for the whole validation, even if our product is pharmacopoeia official, for both API part and FP part. And some of the countries don't ask validation for API part if our product, if the API is pharmacopoeia official. Now, product information and labeling. This is one of the important part for submission of our product. Means whatever the content we are adding in our labeling must be correct and must be according to the labeling guidelines that are given in the respective uh, export country. That means and the whatever contents we are adding in our some SMPC and must comply with our product PIL also. Contents of the SMPP and PIL should be same. And for the labeling, some of the important points that has to be addressed while making our labels are our product, like if it's prescription only medicine, we need to mention POM on the front of our label. And if our pharmaceutical formula, finished formulation has some of the preservatives like sodium paraben, sodium propylparaben, sucrose, or sodium benzoate or sorbitol, we must comply with the statement that has been given in the EMA guidelines. If our formulation is having any of these excipients, then we must check that EMA guideline and we must add the labeling condition or labeling statement that is given in this guideline. And if our product is like eye drops or anything like that, then we need to add the disposal statement that if product is unused or whatever is used, means a disposal statement like any unused product or this material should be disposed of in accordance with this requirement. And for oral liquids and like suspension of serious, in-use stability statement is compulsory to add in our label. And in-use stability statement that is must be after opening use within this many of days, and if you are unused, then it expiry date. Shelf life is another thing, and in-use is another thing. Shelf life can be of 2 or 3 years, but in-use statements are after opening a bottle, use within 30 days, use within 15 days. These are some of the labeling conditions that has to be compulsory, and if our formulation is any of liquid form. That is, once the bottle is open, it should be used within a month or 15 days, 30 days. Then, whenever we are preparing a label, we need to reserve an area for a registration number, which we will get after our product is approved by the authority. And apart from SPC and PIL, a package insert should also be made and should be submitted, and it also comply with the same content that we have given in SMPC and PIL. And SMPC and PIL should also be included in commercial packs. So these are some of the common labeling instruction that we need to follow while using our while making our label. Then we'll come to stability part. Here, stability part is also a very important factor for submission of our dossier, because as I said earlier, our stability data will be according to the product we are going to, will according to the stability zone, and that falls under according to WHO stability zones. So stability data and stability protocol of the finished product, what the stability study include. Stability study should include the testing of those attributes of the finished product that are susceptible to change during the storage and are likely to influence the quality, safety, and efficacy. Means those tests that are going to influence of our finished product are need to be added in our stability data. And stability has to be performed on each individual strength. So if our product is having different strength, but stability data has to be performed on each and every strength of our finished product. And on the conclusion of our stability data, a shelf life must be proposed or concluded with the storage condition. And generally 3 consecutive batches data is required to be submitted. A pilot scale batch is generally 1/10 of a full production scale. Of course, the stability data has to be done on the pilot scale batch or 1/10 of the full production batch. Now here I have given some common stability conditions that has to be followed while performing accelerated stability and long-term stability. Two types of stability data has to be submitted. First is accelerated, that is done for 6 months, and second is long-term, that has to be done for 12 to uh, this is for API. So API can be done for 12 months or 6 months or as per our shelf life of our API. So long-term stability condition varies from 25 to 30 and humidity from 60 to 75 and accelerated from 40 and to 75 humidity. And this is a table that I have taken from WHO site, WHO official guideline chapter, and the link of the same is also added in this presentation. So for elaboration, so for more detail, we can go, you can access that link and then have proper overview of the this stability condition. These are only the normal API stability condition for long-term and accelerated point of view. For FPP stability condition, it's also the same. FPP is also done for both long-term and accelerated. Accelerated is for the finished product is done for 40 to 75 condition, and long-term is done ranging from 2560 to 3075. Why these are different? Because these come under different climatic zone, and we will and what are the different climatic zones we will study here. There are different four type of climatic zone. Zone 1, 2, 3 and zone 4 is divided into two parts, 4A and 4B. Zone 1 is a temperate zone, zone 2 is Mediterranean zone, zone 3 is hot dry zone, zone 4A is a hot humid tropical zone, and 4B is hot and higher humidity. That means we need to follow the zone of which our country is comes under that zone. That means if we are going to export for Tanzania, then it comes under zone 4B. So we need to comply the stability conditions that are mentioned in zone 4B. Uh, Here are some of the long-term stability testing conditions that is divided according to different zones. And long-term conditions, the minimum duration of long-term condition is 12 months and it goes from 2 to 3 months depending upon the shelf life of our finished product. Uh, so if our product comes under if you are going to market in Tanzania, as I said, it falls under zone 4B. So we need to carry out stability condition in 30 and 75. Long-term stability conditions will be 30 or 75. And if we going to file in Nigeria, it comes under zone 4A. So we need to do stability data and with long-term condition having 30, 65. And as I said, this is a link of that WHO guideline document through which we can have different types of stability conditions. We can go through that. And these are normal, intermediate and accelerated stability condition. As I said, accelerated condition come from 40 to 75 and intermediate falls under 30 to 65. And the minimum duration for accelerated and intermediate are 6 months. And if our product has to be stored in refrigerator, we also have different stability and temperature guidelines for a refrigerator and a freezer product also. Now, what are the labeling statements for FPP product? That means if our if our product falls falls under 25 to 60 stability condition, then we need to give a labeling condition like do not store about 25 degrees. This is very important labeling condition. Labeling statement that we need to add in our SMPC or PIR or packaging insert, because it depends upon the whatever the stability zone we are choosing. We have done our stability. If it is comes under 3065 or 3075, we need to give labeling statement like do not store above 30, do not store above 30 degrees Celsius. If it's in refrigerator, we need to add a statement like store in refrigerator or store in freezer. Now moving forward to one of the important topic for submission of our dossier, that is BA/BE studies. BA/BE study, that is bioavailability and bioequivalence studies. Well, the importance of BA and BE studies required, why they are required. So they are required so that our generic product is therapeutically equivalent and interchangeable with our innovator product. Means of our product, if it is generic product, then we need to prove that our generic product is therapeutically equivalent with that of our innovator product. So bioavailability and bioequivalence study are carried out for a new formula, that has to be carried out for both new formulation also and for generic drug also. And to market our generic drug, BA/BE studies are required for the approval from health authorities for very regulated and semi-regulated market also. And drug that has been tested is studied against the well-known or commercial available drug and or innovator drug at same concentration under the same conditions. That means our BA/BE study should be carried out either on the innovator product or any commercially well-known product, but under the same conditions concentration. That means it should have a same strength or having same concentration and under the same conditions also. So what is a bioavailability? Bioavailability refers to the relative amount of the drug from a administration dose which enters the systemic circulation in a rate at which the drug appears in systemic circulation. That means how much amount of our drug is absorbed in our circulation. All the pharmacokinetic parameters are carried out or studied under bioavailability studies. Relative bioavailability, there are two types of bioavailability studies: relative and absolute. Relative is a systemic study that is done after our drug is administered to only orally to that of a oral standard. Means and absolute, that means our active drug is administered to non-intravenous route and it is compared with the same drug administered intravenously. Absolute having the drug route by intravenous and relative having drug administered orally. And here all the pharmacokinetic parameters are discussed, are studied. What is bioequivalence? Equivalence means at what extent and what rate our drug is absorbed and is and are not statistically significant different from those of the reference product when administered at same molar dose. That means if our product is of 10 mg, then we have to have innovator product of the same strength, and what the extent and what rate of absorption our drug is it is absorbed. We study everything in bioequivalence study. There are three types of bioequivalence study: chemical, therapeutic, and pharmaceutical equivalence. When two drugs having the same chemical substance as active ingredient in the same ratio, it is known as chemical equivalence. Means as we said, bioequivalence is to hide at how much extent our product is similar to that of innovator product. That innovator product has to be chemically equivalent. That means it must have same chemical in same active ingredient in the same ratio, that is 10 mg 10 mg. Therapeutic equivalence, that is are similar with the safety and efficacy. Its safety and how much extent its safety and efficacy is similar between generic and innovator product is studied under therapeutic equivalence. And pharmaceutical equivalence means at how much extent is strength, quality, purity, and content uniformity or disintegration or dissolution studies are same to that of the pharmaceutical equivalent. That means a bioequivalent study is divided into up to how much chemical equivalent is our generic to innovator, how much therapeutical equivalent is our product to innovator, and how much pharmaceutically it is equivalent to our innovator product. And in this practice, there are 4 possible scenarios for a bioequivalence and for generic approval. Means for our generic product, it has our generic product has to be either our generic product should have a same drug absorption profile of that of therapeutic product. That means it's AUC and Cmax. That means whenever we do a bioequivalence study, is AUC and Cmax should almost be same. That is, its drug profile, drug absorption profile should almost be same in with our innovator product. And but either it can be drug absorption studies might differ, but it should be therapeutical equivalent. There are 2 main possible scenarios that can happen in BE study or BA-BE study. Either both is drug absorption studies or drug profile studies can be same, or either there can be little change in their absorption study, but they are therapeutically equivalent. Then the 3 and 4 point are drug absorption profile are similar, but they are not therapeutic equivalent, and drug absorption profile are not similar and they are not therapeutic equivalent. For 3 and 4 point, normally the BA-BE studies are not majorly carried out. They are only carried out if our generic product is therapeutically and pharmacokinetic equivalent to our innovator product. Now, how the BA-BE studies are carried out? Whenever we need to perform a BA-BE study of any of the generic product, we need to first we need to first make a protocol and design of our whole, how the process is going to be done. Then we need to select the proper bioanalytical method, and then with what analyte we are going to perform our BA-BE study. Then what will the bioequivalence parameters that we are going to perform, and what will be the statistical approaches that we will study. So these are the some of the points that are mentioned in that we have to study in BA-BE study. We need to firstly make a protocol. Then we need to decide and analyze. We need to study on what are the parameters that we are going to add in our equivalence study. What will the statistical approaches and analysis we are, whatever we have analysis we have done, we need to add in our format. And what, as we said, what we need to first we need to first decide what the design and protocol. So there are 3 types of design that are normally carried out during BA-BE study. First is randomization, replication and error control, and crossover design. Normally first and third, a random BA-BE study is carried out, or a crossover design is preferred for BA-BE studies. Second one, replication and error control, is normally not majorly used for BA-BE study. Here, and what are the inclusions of BA-BE study? When we are going to do a BA-BE study, then what are the other parameters that we need to consider, and how it is done? So for BA-BE study, normally male healthy volunteers has to be elected for performing the study, and whatever the predictive of clinical outcome, that is safety and efficacy of the drug product and clinical trial, has to be mentioned. Bioequivalence study is often conducted utilizing the crossover design or random study that allows comparison within individual subjects, that is, each subject at his or her own control based on pharmacokinetic data collected. Bioequivalence can be accessed using varied statistical method according to some pre-specified regulatory criteria for bioequivalence, and these are indicated by the FDA. An approved generic drug product has to be used as substitute for the brand name drug. That means, first of all, for conducting a BAPE study, the healthy male volunteers has to be elected, and it has to be done on in comparison with a good innovator or vanguard product. And all the pharmacodynamic data are collected according to the design that we have elected. Either it can be crossover design or random design. And all the statistical method has to be mentioned in our study in comparison to the branded name drug. Here a properly filled and signed original copy of BTIF form with all its annexures, uh, including a copy of CD-ROM must be submitted to the pre-qualification of medicine programs together with bioequivalence part of the dossier. That means whenever we are performing a BA study, obviously the BE studies is having a it will be of very big. It will have very much data. So for that, to compensate that, we have to fill a small BTIF form that can be obtained from this link. We have to, this BTIF form consists of all summary of all the data, all the statistical method that we have performed, and we need to fill each one of the list, each one of our summary of each one of our analytical method that we have performed, and then submit to the respective authority. Now, after BA study, the another important topic is biopharmaceutical classification system, that is BCS system. That means it is a comparison between the dissolution profile of our generic drug and innovator drug. Dissolution profile and solubility profile is permeability of for the intermediate release dose or solid or product drug. BCS classification is divided into 4 types, that is class 1, class 2, class 3 and class 4. Class 1 are high solubility, high permeability. Class 2 is low solubility and high permeability. Class 3 is high solubility and low permeability. And class 4 is low solubility and low permeability. Here class 1, that means high and the biowaiver application form is only done on the for class 1 and class 3 products. Class 1 are having the high solubility and high permeability, and class 3 is having high solubility and low permeability. Only the drug having high solubility can come under biowaiver application or biowaiver application form. For class 2 and class 4, a biowaiver application is not falls under that. So basis on, so here as I said, a biowaiver means that is in vivo bioavailability study or bioequivalence study that may be waived and not considered for the product approval. That means bioequivalence study can be waived if we give a biowaiver application form, fulfill biowaiver application form. And bioequivalence study can only be waived, as I said, if our product falls under class 1 and class 3 of BCS classification. If our product falls under class 1 and class 3, we need to perform the comparative resolution profile between 3 media, that is acidic media, that is pH 1.2, and phosphate buffer media, that is 6.8, and pH 4.5. Then normally these are 3 pH mediums that we need to perform our dissolution profile against our innovator product. Then we need to fill the biowaiver application form, that is having the comparative resolution data between generic and innovator product, and then we need to submit the same. And for the products coming under the class 2 and class 4, we need to give bioequivalence study. As I said, there are certain requirements for biowaiver studies. These are like dissolution test in 3 different media, right? As I said, pH 1.2, pH 4.5, and pH 6.8. Dissolution data, dissolution profile in 3 different medias has to be performed between our test product and generic product. 12 samples or 6 samples, either it can be 6 or 12 samples for each media has to be tested, and depending upon the dissolution parameters, that is our dissolution apparatus. That dissolution and our dissolution method can either be taken from each of from the pharmacopoeia if it is official. If our product is official, then dissolution method will be already given in pharmacopoeia. We need to comply with all the pharmaceutical dissolution parameters that are mentioned in pharmacopoeia, and we need to perform the same between our innovator product. And sampling timings can either be 10, 15, 20, 30, 45, and 60, depending upon the nature and the method we use for our finished product. The profiles for the test and reference products must be similar in all three media. That means if we are taking dissolution time for 60 minutes and dissolution apparatus parallel, then it must be same for both product, reference product, that is innovator product, and test product. And for the product that comes under class 1, that is high permeability and high solubility, it is compulsory that within the first 15 minutes, it must dissolve 85% of the total drug. Then only it will comply under class 1 of the dissolution profile. And what we check in dissolution profile, the second thing we check on the dissolution profile is F2, that is similarity factor. It must be more than 50. If it complies with both these parameters, that's F2 and dissolution percentage, then we can say the product is acceptable for biowaiver application. Exemption for biowaiver application, that means what are not included means in biowaiver application, that is reduce the severity of an incorrectly applied biowaiver narrow therapeutic that are excluded. That means that drugs that fall under the narrow therapeutic index drugs, that means they have very narrow solubility form, that are not included in biowaiver application. And some of the pharmaceutical dosage form like oral cavity lozenges and are not applicable for biowaiver application. So these are the this is a summary for biowaiver based conditions that is that they have listed according to EMA, FDA, and WHO. Each one of them have their own set of classification, own set of approval system if we want to apply for biowaiver application. And this is a small summary of the same. That means API should come under BCS class 1 and class 3. If we are going to file our biowaiver application in EMA, then our API should come under BCS class 1 and 3. For FDA, API should only come in BCS 1. If 3, then they will not allow. For WHO, class 1, 2, and 3. For class 2, they have only limited sets of API that is listed in their website. For Excel, they have given the same for excipient, drug type, and drug formulation. And what are the comparative in vitro tests that are required for each one of them? They are listed here. That is for EMA, pH 1 to 1.6. That means we need to give dissolution profile comparison for each one of the media. For FDA, that is 3, and for WHO also for 3 different media. And what are the accepting criteria that are listed here for each one of the regulated market. More information for the BCS application for biowaiver studies can be obtained from this link. And like BTIF, we also have biowaiver application form, and the same has to be filled and submitted to the respective regulatory authority. And the sample of the same can be obtained from the given link. So these are the common requirements for submission of our dossier to any of the regulatory market, starting from its API part to active part, excipients, what are the stability guidelines condition, what are labeling requirements, and what are required for the BAP study. So Ma'am,
Yeah.
do you have any questions?
Thank you, Kinjan, for the presentation. Anyone having any questions?
What is export certificate? There is no any export certificate. That means it's not any particular export certificate. We need to just yes, export certificate. It is not called as export certificate, but it is like application form. Means whenever we are submitting our dossier, each one of the regulatory authority has its own application form. First, we need to fill that application form. We need to sign and stamp from the FPP manufacturer. We need to submit the same. They will go through all. That means it will have some basic summary of our formulation. That means what are the shelf life, what is the brand name, what is the trade name of our product, what is general Q&Q we have used, what is the shelf life. That application will have the small summary of our finished product, and also will have the what is our API manufacturer name and site address, and we need to submit that, and then they will give approval to the one. That means that is an application form that we need to submit, that we need to fill and submit. So that has to be signed and stamped. The regulatory bodies check whether these studies will perform or not with consent. Yes, sometimes means if they have any confusion, or if they find any data to be generated or not official, they can give us a query, and they can ask for a official. They can ask for the what we can say direct contact with that CRO. Then they can check if our bios are perform or not. That only depends. That can be only depend upon the what the regulatory authorities think while evaluating our BAB study. For Mozambique, when the information is not applicable, is understood not required, but in most of the documents is required as mandatory by. Yes. Jayanti ma'am, I'm sorry, I'm not getting your question.
Kinjan, you have any query on this? On
No,
this
actually,
question?
yes, Yayanti ma'am has posted one question, but I'm not getting what she want to ask me. What does it mean? Means any other questions? My Togo is very stringent regarding the I'm sorry, I can't answer why Togo authority is very stringent regarding the registration of product. They have their own set of guidelines, and they only see whatever documents are perfect or not, and they give approval on that according to our documentation only. Yes, in Nigeria, it is compulsory to provide LOA from from API manufacturer. Yes, it is compulsory. If you are having that third-party contract manufacturing, then it is necessary to give the LOA certificate, and also and also the GMP certificate is also important, and LOA certificate is also compulsory. Yes, ma'am. Regarding that not applicable question, yes, if it is written in, but if but we have to see that if our product is generic or not. If our product is generic, then and if it comes under not applicable part, then we no need to add in module 1. And what is our drug? What is our finished product? It depends on that. Question.
Hello, ma'am.
Yes.
Ma'am, I want to ask. Now there is the requirement for IVC guideline for Module 2 QoS model. So before that, before coming to that, there was no requirement like that, and our previous Module 2 was मतलब वो जो सारे जो पॉइंट थे वो कवर हो रहे थे, तो अभी उन्होंने सिर्फ टेबल्स ऐड किए हैं, तो अभी उसमें सिर्फ टेबल में सिर्फ भरना है, ऐसा करना है क्या?
हाँ, now अभी most of the countries have changed their module 2 format. Most of the they need QAS, that is quality overall summary, and QI is quality information summary. QIS has to be submitted in module 1 and QAS is module 2. So before QAS, what we are doing, that we need to give each and every summary of API and FP in our module 2. But now they have only come with quality overall summary. That means we just need to fill the QoS. We need to fill what are information that are listed in QoS, and we need to submit. We don't need to add any other documentation except QoS. For many countries, they only need QoS in module 2, FP part.
Okay, thank you, ma'am.
Anyone else? I guess we are done for the session. Okay. Thank you, Kinjan.
Thank you so much, ma'am, for having me here.
Thanks, everyone. Thank you.